Hello. I hope that everyone is safe and well. My name is Lobbezoo, and I'm the Product Support and Development Manager at Astrea Bioseparations Ltd, formerly known as Metris Bioseparations Ltd. I've been with the company for nearly 16 years, and in that time, I've performed downstream processing, troubleshooting, and working with our products and customers alike. In this presentation, I want to talk to you about the turnkey approach that Astrea offers in delivering GMP-ready adsorbents and columns, starting from initial ligand discovery, thereby supporting our customers and fulfilling their unmet needs. One of the biggest challenges the biotech and biotherapeutic companies are facing is the development of processes that deliver safe, cost-effective, and efficient products. The main issue is that biotherapeutics require a significant amount of effort to be committed to their purification process and frequently on the removal of impurities from their product arising from the complex feedstocks. Multiple chromatographic steps are therefore used, but in more and more cases, mixed-mode chromatography, and in particular affinity chromatography, is being more frequently used for these purposes. And unless there's an off-the-shelf affinity product, which for specific novel therapeutics is unlikely, the use of affinity chromatography requires the development of novel ligands and adsorbents specific to the target. Astrea Bioseparations is the only bioseparations company to offer ligand discovery and adsorbent development services in combination with delivering large-scale manufacture of chromatography adsorbents and production of pre-packed chromatography columns to GMP standards. In this presentation, I will cover the strategies we take and describe a recent case study whereby we've supported a customer from ligand discovery through the development of an adsorbent delivered in a pre-packed column format, ready for them to use in manufacture. To support our clients and customers with delivering their requirements, we have a dedicated program or roadmap of work and processes. The process consists of 3 discrete phases, and I will go into detail on each of these phases later on. At the start of the project, using computational chemistry, a number of suitable ligands are identified. Then, in phase 2, an adsorbent is selected and developed in parallel, alongside the identification of suitable conditions and operational parameters. This culminates in pinpointing a final adsorbent and process, which is ready for phase 3. This is where the adsorbent is transferred to manufacturing, accompanied with a full regulatory support package, and thus supplying a custom adsorbent or, if required, a pre-packed column to enable the customer to deliver. The first phase is to identify ligand candidates specific to the target and successive screening to narrow in on the top candidates for adsorbent and process development. So how do we decide what route to take? What we know about the target and its interactions help us decide which strategy to follow for ligand discovery. If the target protein structure is known and available, and there is a known binding site, we can use ligand docking and virtual high-throughput screening techniques. Ligand docking means that we take selected ligands from our virtual library and virtually place them in the binding site and evaluate the fit of the ligand within the binding site. Alternatively, if the binding site is not known, we will use the binding site determination or blind docking to identify ligands suitable for the target protein. This is where the virtual ligand library is screened in silico against the protein to identify ligands that have an affinity for the protein binding site. When we don't have structural information about the protein, we can use information about known inhibitors and search for ligands with structural similarities. Of course, there are also occasions when there is no structural information available or known inhibitors to the target protein. The ligand discovery approach then is via the systematic high-throughput screening of a selection of libraries or the identification of a hit through a more discrete but diverse library. As mentioned earlier, I want to describe a case study whereby we supported one of our customers in supplying them with a custom affinity adsorbent. In this case study, we already knew the structure of the target protein. We were also aware that there were several different isoforms of the target protein, which the customer ideally wanted a ligand which could bind to all, i.e. a platform technology. The feedstock which was supplied was a clarified cell culture supernatant containing the target isoforms but at low concentrations. This added an extra layer of complexity. Regarding the ligand discovery strategy, the following path was selected as the target protein structure was known and available. However, the ligand binding site was not. A ligand binding site had to be determined, followed by docking studies using computational chemistry. As we wanted a ligand that could be used for multiple isoforms of the target protein or proteins, we started by determining common clefts accessible to the ligands. The surface of the protein was scanned for possible ligand binding sites, and we used docking studies to dock the ligand structures to the possible binding sites, as well as determine the binding energy. Overall, the more negative, the more favorable. However, a balance is needed to be struck, whereby we obtained a good fit for binding, i.e. selectivity, but not too tight as to not be able to recover the target material using conventional elution conditions. From these studies, we selected a number of possible candidates. However, we needed to consider many factors. Ligand orientation is one, as well as certain restrictive factors, which can include cost, ligand availability, the ligand toxicity, as well as physical and chemical restrictions such as site accessibility and the spacer arm which attaches the ligand to the adsorbent. The restrictive factors, such as the spacer arm, could have both a positive or negative influence on the binding. If the spacer arm is too hydrophobic, there could be repulsive forces which could detrimentally affect the binding. Or conversely, certain charge groups or the length of the spacer arm may actually aid in the ligand docking. Based on selected structures from the virtual screening, we designed a library of trizene-based compounds. The library explored the chemical space around the selected structures. It is useful to constrain your library so as to focus the screening efforts. On this slide, we have depicted the same binding site, but two different candidates with different structures. It is worth noting that the positioning or orientation of the ligand within the binding site is important to allow the spacer arm to be attached. This is indicated with the circle. In both cases, the ligands have an NH3 group to couple onto the spacer arm in an acceptable position. It is worth highlighting, of course, that in some cases the protein structure is not known. In these cases, work can be done by initially exploring a wider chemical space, then narrowing down the focus as the results are generated. This is more of an iterative approach. A lot of time is spent during this phase to ensure that the right targets are selected for generating suitable libraries. This approach allows for the synthesis of combinatorial libraries, which are ideal for identifying ligands with mixed modes of action. The libraries generated are with trizene-based ligands, which is the basis of most of our ligands. Trizene ligands are ligands which are small chemical constructs developed on a core trizene ring attached via a spacer arm onto an agarose or other support. The ligand is built up by activating the dichlorotrizene, then coupling a primary amine to the trizene core and creating an intermediate. The monochlorotrizene intermediate is then further substituted to a final two-dimensional ligand. Astrea standardly create an 8x8 array library of subtly different ligands. This building of ligand libraries using a wide range of different chemical compounds allows for the creation of an extensive library incorporating a wide variety of properties which are caustic stable. The libraries generated are puriplates, which are comprised of 64 discrete packed microcolumns with top and bottom frits and reservoirs above. These columns will drain freely under gravity, so automated liquid handling systems are not required, only a multi-channel pipette. These puriplate libraries allow us to perform the screening like using a standard column, allowing for equilibration, sample application, wash steps, elution, and CIP steps. The fractions of interest are collected by drainage into deep well collection plates, which can be analyzed using discrete analytical tools such as SDS-Page, or by a variety of plate-based assays to measure the amount of target protein or impurities in each fraction. This allows for a variety of factors to be identified for the suitable ligand which binds to the target. Once the screening is complete, the fraction samples are tested using a range of assays. The data can then be presented as a heat map, such like here. This heat map can be built up based on predetermined criteria such as binding, recovery, purity, ease of elution, or others. From these criteria, the best adsorbents are selected. A heat map can have a nominal scoring of 1 to 5 when the factors based on factors which don't have a clear defined value assigned, such as ease of elution. Or there could be a percentage value out of 100, whereby the higher the value, the better. In this case, the poor performing ligands are colored in blue, and the best performing ligands are colored in red. From the screening, we select one or more adsorbents for further development, or we design a new library based on these results and repeat the process. In this case, as there were several high-performing candidates, additional scrutiny was applied to the results to identify the top candidates in order to proceed to the adsorbent development. Once the first phase is complete and one or more, usually up to 6 adsorbents, depending on what was identified during ligand discovery, and these candidates have been identified, the next phase begins: adsorbent development and process development. The adsorbent development can be effectively divided into 2 parts: the chemistry development and the downstream processing. The chemistry development focuses on many aspects, including the optimization of the spacer arm, the length and how hydrophobic or how hydrophilic it needs to be, as mentioned earlier, the ligand density, investigating a range of ligand density adsorbents to explore selectivity and capacity, ligand attachment chemistry, the base matrix selection. I previously mentioned that we couple the ligand to PureBead, which is our near monodispersed agarose base matrix material. However, other base matrices or even other supports can be investigated. Rational optimization of the ligand and adsorbent synthesis for improving cost effectiveness and process robustness is also carried out, and the list continues for the chemistry development. The other part is the downstream processing. This focuses on the development of a suitable wash, elution, regeneration, and CIP conditions, as well as testing the performance of the adsorbents under the standard process conditions. The downstream processing will perform all the verification work in relation to the work which was carried out throughout the chemistry development for the ligand and adsorbent synthesis. The adsorbent development uses scaled-up synthesis of the ligands, which are packed into a variety of larger columns and are tested for performance. There are There are a range of key factors at this stage, including, as said, binding capacity, selectivity, elution conditions, total protein binding, impurity profiles, and recoveries. CIP regimes are also investigated to ensure that the adsorbent is stable and reusable. In this case, the example here is after the optimization and the development. On the right-hand side is the STS page of the ligand density optimization. We identified that a lower ligand density was preferable not only to improve the purity, but also to ensure good recovery. It's worth noting that although this ligand showed high selectivity for the target protein, certain charge groups present on the ligand did cause some minor non-target binding, which was accentuated with the higher ligand densities. Sometimes there is a trade-off between purity and capacity. It's worth noting on the left, where we see the material in the load and the final target in the elution, we achieved 14 grams per liter binding with a 96% purity and a 98% recovery after the development and optimization. Once the adsorbent has been fully developed, 3 verification batches are produced, usually at the 1 liter scale, and are evaluated for performance and consistency within the limits of the process. The process samples in this case study were run on an SDS page and compared between the 3 batches, demonstrating high comparability and elution fractions with high purity across the single step. Once the adsorbent development is complete, the adsorbent is ready for tech transfer into production at our manufacturing facility on the Isle of Man. During tech transfer, all the documents and reports required are transferred to our manufacturing site via our dedicated TT team. A single batch of adsorbent is produced at the same or larger scale than the verification batches. The TT batch is fully evaluated and assessed for comparability to that of the verification batches. For the case study, an STS page of the tech transfer batch was performed and demonstrated comparability to the verification batches, with a strong main target band visible. Once the tech transfer batch has been fully evaluated, manufacturer will commence creating at least 3 validation batches. During tech transfer and at the start of manufacture, the equipment requirements and user requirement specifications are evaluated. The incoming raw materials, in-process assays, and final product assays for the product are assessed for their suitability and validated. Ligand and adsorbent synthesis methods are validated, and all required standard operating procedures and batch manufacturing records are established. Validation of the adsorbent synthesis is usually performed with the manufacture of a minimum of 3 batches, usually at a larger scale to that of the TT batch, demonstrating comparability and quality. The custom adsorbent is now a fully validated and supported product and can be supplied to the customer. However, to fully support and enable the customer, Astrea also offers a column packing service. If this is desired, a packing development program is initiated to fully understand the packing properties of the adsorbent and define all the parameters required to produce a uniform and robust packing process. This method follows the same route once again, back through tech transfer and into manufacture. Along with our external testing, including transportation testing, we ensure that we can deliver a plug-and-play product to the customer. Astrea also provide smaller lab-scale columns to perform screening and process development with their custom adsorbent if selected, or one of a range of off-the-shelf products. For the case study discussed in this presentation, we provided the customer with multiple batches of pre-packed Evolve D 7 and 10 centimeter diameter GMP-ready columns, which are a perfect fit for their requirements. We support our customers through each stage, from ligand discovery to adsorbent and process development. Once a suitable candidate has been identified and optimized, we undertake the tech transfer of a robust process for either adsorbent or column packing services. We also provide a range of columns from lab to process scale. Through our technology, people, and our processes, Astrea has the capability to provide a product or service which fits your needs. If anyone wants to find out more, please visit our website at www.astreabioseparations.com. Thank you for your time and attention. If you have any questions regarding this presentation or any of our products or services, please feel free to stop by our booth or email sales@astrea-bio.com.